Hello everyone, and welcome to our lecture on microbial growth and nutrition. Like with microbial metabolism, this is probably one of my favorite subjects to talk about. And I know it's not everyone's favorite subject to talk about, but hopefully I've been able to put all of this information into enough of an interesting context that you can at least appreciate why it is significant and important. This is a fairly information-rich chapter. It has a lot of vocabulary terms, a lot of definitions, and there are a lot of objectives, so this is one you may want to go back over a couple of times to make sure that you've got all of the information understood correctly, you've got all of the definitions, and you're ready to move on to other lectures. In our last lecture, we talked about 2 out of the 3 things that microbes need for metabolism to happen. We talked about enzymes, which are protein molecules that help catalyze chemical reactions inside cells or help make those chemical reactions occur. And we talked about energy. And energy is the ability to do work or cause change. The third thing that microbes need to be able to carry out metabolism are nutrients and raw materials. So to start us off, just to define what nutrients are, this is another term that a lot of times we think we know what it means, but we don't have a full understanding of it. So just to make sure we all have the exact same definition of nutrient, a nutrient is a chemical that is taken from the environment and used for growth and metabolism. When we talk about nutrients, there are lots of different ways we can categorize them or divide them into categories. And 2 of the broadest categories or ways to sort out nutrients are to look at whether an organism has to have a certain nutrient to survive or doesn't have to have a certain nutrient to survive. Nutrients that organisms must have to survive are called essential nutrients. And from there, essential nutrients can be divided into 2 more categories called the general essential nutrients and then specific essential nutrients. General essential nutrients are those that are common to all living organisms. And a good example of those are the sponge elements: sulfur, phosphorus, oxygen, nitrogen, carbon, and hydrogen. Every single living organism on the planet, whether that is a microbe, whether that's a person, whether that's a plant, whether that is an animal, every living thing on the planet must have all 6 of these elements as part of their nutrients, or they will die. The reason for that is these elements make up the macromolecules. They make up carbohydrates, proteins, lipids, and nucleic acids. And without those, organisms simply do not exist. Most of the time, general essential nutrients are also what we call macronutrients, meaning an organism requires these in large quantities. The reason for that is since most cells are made up primarily of things like lipids, carbohydrates, proteins, and nucleic acids, organisms need these 6 elements in large quantities so they can make those macromolecules. Our other category of essential nutrient is the specific essential nutrient. These types of nutrients may be required by one type of organism, but not another type of organism. And the reason for that, the reason that there are specific essential nutrients for every individual type of organism on the planet, is because all organisms are slightly different from other ones. So I've got some listed here. You've got some examples that I've listed out. There are obviously many more than this, but we've talked about the diatoms before, which are certain types of algae that have cell walls made of glass. And so for those organisms, it's very important that they have a sufficient supply of silicon so that they can make their cell walls, because this element goes into making their cell walls. Many organisms require copper or iodine. Another good example is iron or manganese, etc. And the gist of this concept is that while all organisms have to have the general essential elements, every individual type of organism or every individual species of organism will have their own unique set of specific essential nutrients, because all organisms are unique as well. So while all living organisms have certain things in common, All living organisms also have their own specific need for certain types of nutrients that they require from their environment. Oftentimes, although not always, the specific types of nutrients an organism requires are micronutrients. And this means that though an organism needs this nutrient to survive, it needs it in small quantities. So just as an example in humans, there are lots of different metals that we require for things like cofactors and making sure our electron transport chains work, etc. And although we need these metals, we have to have them to survive. We need them in small quantities. If we get too much of that inside our body, it actually is very, very damaging. And this type of concept is true for other types of organisms as well. You can provide, let's say, bacteria in a lab with iron that they might require, but if you overload them with iron, that can actually be very damaging to their cells and could even kill them just as quickly as not having iron. So these types of nutrients, while specific essential nutrients are required, they oftentimes are micronutrients, meaning an organism needs them in smaller quantities, or that could be very damaging. The last thing I want to mention on this page are growth factors, which are technically types of specific essential nutrients. Whether they're needed in large or small quantities can vary. These are called growth factors, and growth factors are relatively large organic molecules that organisms have to be supplied with basically in a pre-made fashion to be able to survive. So I'm going to give you an example from the human body because this will make sense to everyone. But for humans, vitamin C is technically a growth factor. This is a relatively large organic molecule that we must take in with our diet somehow. We have to take it in from the environment in a pre-made form. If you give someone no vitamin C and just give them the components to make vitamin C, that will not work and they will eventually die. So for humans, one of our growth factors, we have many, but one of them is vitamin C. And this is something that we just have to get from our environment in a pre-made form. We can't synthesize it in our bodies, even if we have all of the raw materials for it. We have to have it pre-made, and if we don't have it, we will eventually die. It'll take a while, but eventually it will lead to death. And lots of organisms, especially those that infect humans and animals, also require growth factors. And many times, the growth factors that they require are actually ones that we make, are actually chemicals that are found inside our bodies. And so many organisms are highly evolved to be what are essentially very good parasites to the human and the animal body because they require certain growth factors, and our bodies happen to have those chemicals in large amounts, meaning we are a very good environment for those types of microbes to grow in. In addition to looking at what types of nutrients an organism will need, another thing that is important for us to understand about microbes is where they get their carbon and where they get their energy. The reason it's so important to understand this is because it tells us a lot about the environment this organism or any given organism could live in. And especially when we're talking about the healthcare fields, we want to understand whether an organism would be adapted to live in a human or animal body or not adapted for that. Organisms that are adapted to live inside a human or animal body would be very good, or at least potentially very good, at being pathogens or microbes that can infect us and cause damage, whereas organisms that are not adapted to live inside a human or animal body are probably not going to be pathogens. So to understand the type of environment that an organism would grow best in, we look at two things. We look at their source of carbon and we look at their source of energy. There are two very broad categories of carbon compounds. Those are organic sources, and then there are inorganic sources. Remember that organic molecules have carbon and hydrogen at a minimum. They often also have oxygen, not always, but often. And inorganic molecules are those that may have carbon or may have hydrogen, but do not have both. So organisms that require their carbon to be in organic form are called heterotrophs. Organisms that require their carbon to be in inorganic form are called autotrophs. Heterotrophs are better at infecting humans and animals than autotrophs. The reason for that is that our bodies are excellent sources of organic carbon and less good sources of inorganic carbon. Things like carbohydrates, lipids, proteins, nucleic acids, all of these things that make up the human and animal body are sources of organic carbon. Now, our bodies typically do have some inorganic carbon in them as well, especially in the form of something like CO2. But overall, our bodies don't have a lot of inorganic carbon-containing compounds in them, and so they are not an ideal environment for autotrophs to live in. The next thing we look at when we're trying to figure out where an organism might live is the energy source. And energy sources typically come from either light or chemicals. Organisms that use light as an energy source are called phototrophs, and organisms that use chemicals are called chemotrophs. So here you have an example of light, and these are sugar cubes because carbohydrates or sugars are some of the most common chemicals that are used as a source of energy. Humans and animals are not really great sources of light, and so typically the organisms that can infect us are not phototrophs. However, we are a great source of chemicals, and so the organisms that do infect us are typically chemotrophs. Now, to make things a little bit more complicated, we can actually put these terms together, and we can call something a chemoheterotroph, a chemoautotroph, a photoheterotroph, or a photoautotroph. So I will write those out, and then we will talk about which type is most likely to infect us. A chemoheterotroph is something that uses chemicals as a source of energy and uses organic carbon. A chemoautotroph is something that uses chemicals as a source of energy and an inorganic source of carbon. A photoheterotroph would use light as a source of energy and organic carbon. And finally, a photoautotroph would use light as a source of energy and inorganic carbon. So out of these 4 categories of organisms, and there are actually more, it does get more complicated than this, but for our purposes, we're just going to say that all organisms on the planet really fall into one of these categories. And out of these sources, photoheterotrophs and photoautotrophs really don't infect humans or animals because, again, like I mentioned, we're really not great sources of light. We don't have the right kind of energy. Even if we have the right type of carbon, we don't have the right type of energy because we don't produce light, really. So chemotrophs, whether they are chemoheterotrophs or chemoautotrophs, are much more likely to infect us. And out of those, the chemoheterotroph is the type of organism that most of the things that infect us are. These are organisms that use chemicals as an energy source, and they use organic carbon as their carbon source. And humans and animals are excellent sources of chemicals and excellent sources of organic carbon. So out of all of the different ways that, or combinations that organisms might get their energy and might get their carbon, the one that is most likely to infect us is the chemoheterotroph, because we provide the ideal environment, really. We provide the chemicals they need for energy, and we provide the organic carbon they need as their source of carbon. So we really provide everything they need for them to be able to grow. Now, of course, not every single organism that is a chemoheterotroph will be infectious. It's just that out of the organisms that are infectious, the vast majority are going to be chemoheterotrophs because we provide the chemicals they need and we provide the type of carbon that they need. Aside from nutrients and energy, microbes, and really all organisms, require some additional conditions to be able to grow. They have to have the appropriate electron acceptors so that they can make ATP, and they also have to have the right environment. Things like temperature, pH, osmolarity, all of those things influence metabolism and cell structure and determine whether or not the organism is able to reproduce itself. So we are going to spend the next couple of slides talking about electron acceptors, and then we're going to talk about things like temperature, pH, osmolarity, and start to understand how those factors can affect microbial growth as well. The big picture to all of this is to keep in mind that, especially for people going into the healthcare professions, it's important to understand what types of factors influence microbial growth, because if we are trying to combat an infection or we are trying to keep a microbe from growing in a certain place, we have to alter, in many cases, some kind of condition that will stop them from growing. And so before we could do that, we first have to understand how they do grow, and then we can start to understand how we can interfere with that growth in a clinical or medical setting. You all know a little bit about this already because we talked about electron acceptors a little bit in our previous lecture when we were talking about ATP production. But just to review that and maybe go into some more depth about that, electron acceptors are molecules that accept electrons during ATP production. And if an organism is using respiration to create ATP, whether that is aerobic or anaerobic, they are using an inorganic electron acceptor. And if they are going through fermentation, they are using an organic electron acceptor. We oftentimes categorize microbes or evaluate their growth abilities based on what type of electron acceptor they use, and especially whether or not they use oxygen as a final electron acceptor to create ATP. So oxygen is interesting because many people think that absolutely all organisms must have oxygen to be able to survive. But the reality is there are lots of organisms on the planet that use oxygen, but there are also lots of organisms that don't use oxygen to make ATP, and there are some that are even killed by the presence of oxygen. When we're talking about oxygen as an electron acceptor, we can group almost all organisms or all microbes into 5 main categories. The first category are the obligate aerobes. These are organisms that absolutely must have oxygen to be able to make ATP and to survive. If they do not have oxygen, they absolutely will die. So if we were growing these in broth cultures, these types of microbes would grow up at the top here. The reason for that is this is where the oxygen is, and any farther too much below the surface, they're not going to survive very well. Sorry, it's hard for me to draw and talk at the same time. So these organisms absolutely have to have oxygen to be able to make ATP, and if they are not in the presence of oxygen, they will die. They will not be able to create ATP, and they will not survive. They grow near the top of broth cultures because that is where the oxygen is. The next group of organisms are the microaerophiles. Technically, these are a subcategory of obligate aerobes. They do need oxygen. However, they can't tolerate it in high amounts. So they have to have oxygen to make ATP, but they can't have lots of oxygen. They have to have kind of a moderate amount of oxygen. So these will grow near the surface of a broth, but not right at the surface of a broth. The reason for that is at the top, there's just too much oxygen. It's actually poisonous for them, and so they will die. But they need a little bit of oxygen that has diffused into the medium. And right below the surface usually is a pretty good area for them to find that sweet spot where they have enough oxygen to create ATP, but not so much that it will kill them. Our third category of microbes are facultative aerobes. These are organisms that typically prefer to use oxygen to make ATP if it is available, but they can make ATP without oxygen. So they will grow best at the top here. But we can actually find them growing kind of anywhere throughout the medium. They'll be at the highest concentration up near the top because oxygen typically helps produce the most ATP when it can be used, if it doesn't kill something. But these types of organisms will really grow kind of anywhere throughout the broth culture, even down at the bottom, even if it's not in very high amounts. Because if they can't find oxygen as an electron acceptor for ATP production, they can use something else. Either they can use an organic electron acceptor to go through fermentation, or maybe they can find another type of inorganic electron acceptor and go through anaerobic respiration. Our fourth category of microbes are the aerotolerant anaerobes. These are organisms that never use oxygen to make ATP. They may use oxygen for something else, like to make their macromolecules, but they do not use oxygen as a final electron acceptor for ATP production. They will grow best down towards the bottom of a culture because they prefer not to be around oxygen. However, they can also grow in other places in the broth culture, even near the top, even if not in very high quantities, because the presence of oxygen will not kill them. So they can be around oxygen, they're never going to use it, but they can tolerate it. And then our final category are basically the exact opposite of the obligate aerobes. These are obligate anaerobes. These are microbes that absolutely cannot be around oxygen or it will kill them, and they never use oxygen to make ATP. Obviously, if it's going to kill them, they are not going to use oxygen to make ATP. They'll need oxygen to do things like make carbohydrates or make proteins, but they will never use it for ATP production because it will kill them. These will only grow near the bottom of the tube because this is where there isn't really any oxygen left over. So gases typically are poorly soluble in fluids, which means that even though there is oxygen at the surface up here, it really only penetrates probably about down to here really well. And then they might have a little bit farther down here. But once you get down here to the bottom of the tube, there's no real oxygen to speak of. And so these obligate anaerobes can grow here because there's no oxygen there to poison them. Knowing about the oxygen requirements of an organism is very useful when we're trying to understand whether it might infect a human or animal, and if it does, where it might live or how it might carry out metabolic functions. For example, an organism that absolutely must have oxygen, whether that is an obligate aerobe or maybe a microaerophile, would probably live maybe in the lungs or somewhere near the surface of the body because there's a little bit more oxygen available to it. Whereas an obligate anaerobe would probably live deeper in the tissue and do more damage inside the tissues rather than superficially. On the other hand, a facultative aerobe or an air-tolerant anaerobe could potentially live in lots of different environments in the body. And so we would have to be aware of that and make sure we understand that that organism has the potential to infect lots of different locations. There are, of course, exceptions to all of these different places that I've talked about in terms of infections and where it might live in the body. But in general, if you have something that is going to require oxygen, it will probably live near the lungs or near the surface of the body. If something absolutely cannot tolerate oxygen, it will probably live deeper in the body. And then if you have something that can do with or without oxygen, that's kind of the worst case scenario actually, because it can live anywhere and has the potential to spread to lots of different locations. The next environmental factor that we will talk about is the temperature. All organisms have a set of what we call cardinal temperatures. These are a set of 3 temperatures that basically determine where an organism is able to live in the world. The cardinal temperatures are specific for each type of organism. So every species of organism on the planet technically has its own set of cardinal temperatures. And the cardinal temperatures are made up of the minimum, the optimum, and the maximum. The minimum temperature is the lowest temperature an organism can survive at. Growth typically is not happening very quickly, if at all. And if an organism drops below the minimum temperature, its plasma membrane or its cell membrane tends to freeze up. And once the plasma membrane freezes, even if the cytoplasm inside a cell doesn't freeze, it means that the organism or the cell cannot bring anything in from the outside or get rid of anything from the inside. And so that typically leads to death. The optimum temperature for an organism is the temperature that everything happens the best at for that organism. It is the temperature where its metabolic reactions are proceeding the way they are meant to, its enzymes are functioning, its plasma membrane has the right fluidity, so it's not too solid, it's not too liquid, and basically everything is correct for those organisms' proteins to be functioning the way they need to, and the organism to survive. If an organism is going to grow, it typically happens fastest right around this optimum temperature. Now, past the optimum temperature is the maximum temperature for an organism. And as you probably have figured out, the maximum temperature is the highest temperature an organism can survive at. Any higher than the maximum, even if it's maybe only by half a degree or a tenth of a degree, means its enzymes will denature, its membrane will fall apart, and it will die. So remember, like I said, every species technically has its own minimum, optimum, and maximum. For example, let's say humans, our optimum temperature is just about 37 degrees Celsius, at least for our internal body temperature, which is right about 98.6 degrees Fahrenheit. And that works very well for us. But there are other organisms on the planet that would actually, our optimum might be higher than their maximum. And so if you were to put that organism inside our body, it would die because its cells would not be able to handle what to it is a very high temperature, even though that is our optimum temperature. So when we talk about these cardinal temperatures and try to understand how temperature affects microbial growth, we start to categorize organisms based on where their optimum temperature really is. If an optimum temperature is what we would consider, us as humans would consider very cold, that is a psychrophile. Organisms that live in the cold are considered psychrophiles. These are organisms that might grow in the snow or might grow on the sides of glaciers or might grow in frozen soil. So those are organisms that actually live at incredibly cold temperatures. And if you were to try to take them and put them in the human body or in an animal body, they would not be able to survive. So typically, psychrophiles are not infectious to humans and animals because our body temperature is way past their maximum temperature. There is a special subtype of psychrophiles that are called psychrotrophs. Technically, these organisms don't love living in the cold. They're not as adapted as a true psychrophile would be, but they can live in the cold and they can grow decently in the cold. Great examples of psychrotrophs are organisms that you find growing in your refrigerator. When you have something in your refrigerator and it has maybe been pushed to the back and left for a while, maybe because you forgot about it or just didn't feel like dealing with it, and eventually you pull it back out and see something growing on it, that organism is probably a psychotroph. It probably doesn't love the cold temperatures of your refrigerator, but it can survive them, it can tolerate them, and it can grow, even if it grows relatively slowly, in that colder temperature. The next category of organisms are mesophiles. These are organisms that grow best or have an optimum temperature in what we would consider middle environments. A good example of a mesophile is something like E. coli or Staphylococcus. These organisms tend to grow best at just about body temperature of humans, and so that is a middle temperature. These are the ones that are most likely to infect us. They're the ones that are most likely to cause problems when we're talking about something like foodborne illness, where we get microbes in our food and it makes us sick as a result. And so these are ones that we really need to pay attention to and watch out for and make sure that, especially in the environment, we are controlling for them and being careful about them so that they don't get into our bodies or get into our food and make us sick. Thermophiles are organisms that live in incredibly hot temperatures. These are things that would live in something like a hot spring, whether that is a superficial hot spring or a hot spring at the bottom of the ocean. There are things called thermal vents at the bottom of the ocean that many thermophiles will live in. And these ones have incredibly high optimum temperatures. Typically, they require such a high temperature, in fact, some of them can even live at boiling water, that if you were to put a thermophile in the human body would be too cold for that organism to survive in, and it would die as a result. Now, there are subcategories of these, especially for the thermophiles. I am not going to test you on all of the temperatures. I am not going to make you memorize the temperatures of any of them. If you're interested, we can certainly talk about those more through email. I actually have done a lot of work with thermophiles, so they are definitely a type of organism that I am really interested and really like to talk about. However, I am not going to make you guys memorize everything about them because the big thing to know about thermophiles is that they tend to not be infectious because our body is actually too cold for them. The next environmental factor that we will talk about is pH. So remember, the pH scale goes pretty much from 0 to 14. Anything from 0 to 7 is considered a low pH, and that is acidic. Anything at exactly 7 is neutral. And anything from 7 to 14 is a high pH, and that is considered a base. Most organisms on the planet require a pH of somewhere between 6 and 8 to grow. Some organisms do a little bit better towards the 6 range, some organisms do a little bit better towards the 8 range, some organisms need to be much closer to 7. But on the whole, if you kind of take the average of all organisms on the planet, most organisms have a pH range of about 6 to 8 that they can survive and thrive in. This allows for enzymes to function properly, it allows for the cell membrane to stay intact and not fall apart, and basically just allows for metabolic reactions to proceed at the rate they need to for the organism to survive. There are some pretty neat exceptions to this, however, and there are organisms that live on the very extreme ends of the pH scale. Acidophiles are organisms that typically grow at pHs below about 5.5. We can find these in various locations. There are actually acidophiles that live in things like vinegar. So sometimes if you get raw vinegar or unfiltered vinegar, you may have some organisms in there that are technically acidophiles. But out in the environment, we often find acidophiles surrounding mining operations. A lot of times, mining operations use acids to get metals out of the soil. So to be able to get small particles of metal out of the soil, if you lower the pH of that soil with an acid, the metal tends to kind of come to the surface basically, and means you can get it out easier. And so oftentimes waterways around mines will have acidophiles living in them because those waterways are very acidic due to the mining operations. Sometimes those acidophiles can even be helpful in cleaning up that environment a little bit. If that water is very, very acidic and potentially damaging to the environment around it, then acidophiles can occasionally be used in bioremediation operations. Although in kind of the opposite of that, there are sometimes or some occasions when natural acidophiles will make the situation even worse and increase the acid content of a waterway or some soil. On the opposite end of the pH spectrum from acidophiles are the alkalophiles, or sometimes people say the alkalinophiles. It just kind of depends. Either way is technically acceptable. And these are organisms that grow at very high pHs. So these require a pH of at least 7.5, but most of them grow around 9, some around 10, 11, 12, 13. So typically the alkaphiles grow around a pH of 9, but it could be much higher than that as well. And these organisms are found in a couple of different places in the environment. One place we find them are hydrothermal vents in the ocean. I mentioned those on a previous slide when we were talking about thermophiles. So these organisms are very interesting to us for lots of different reasons, but one of them being is they provide enzymes for industrial applications that are very, very stable. We can use their enzymes in all kinds of industrial processes to help keep enzymes working even under what would be normally kind of punishing or damaging conditions. So they are a great source of enzymes for things like biotechnology and industrial microbiology to help us create certain products that otherwise would not be able to be quite as easy to make. Our last environmental condition that we will talk about is the requirement for a certain type of osmolarity. Organisms on the planet have technically a very wide range of tolerance toward different amounts of solute concentrations. And when we're talking about solute concentrations, we're typically talking about salt and or sugar. So there are lots of different solutes that could exist in the world, but most of the time when we're talking about osmolarity and tonicity and stuff like that, we're talking about salt or sugar. Most organisms prefer an isotonic environment or a slightly hypotonic environment because that helps keep water coming into their cells. It kind of helps keep this constant flow of water coming in, not a very hypotonic environment because that would cause their cells to burst, but a slightly hypotonic environment ensures that water and other substances can always be flowing into the cell. So that's for most cells, but as you've probably figured out by now, microbiology has a lot of exceptions and a lot of unusual types of organisms in it. And the two exceptions that we will talk about are the halophiles and the osmophiles. Halophiles are organisms that love very salty conditions, and in fact, not only do they love them, but they require them for their cell membrane to be able to stay intact. If you take something that is a true halophile out of a very salty condition and put it into something like an isotonic or slightly hypotonic environment, it will die. They basically have to live in an incredibly hypertonic environment to be able to survive. Now, there is a wide range of salt concentrations or salinity percentages that people use when they're defining halophiles. And there is some discussion within the community that study halophiles about what constitutes a true halophile or not. But basically, we will just say that true halophiles require salt conditions or salt concentrations that are even higher than the ocean. So the ocean is approximately 3.5% salinity. That can vary depending on where exactly in the ocean you're talking about or what other conditions are going on. But overall, kind of on average, the ocean has a salinity of about 3.5%. And most people consider halophiles those organisms that have to live at concentrations above that. So these types of organisms can't even necessarily live in the ocean because it's not salty enough. These organisms live in places like the Great Salt Lake or the Dead Sea or other very, very salty aquatic conditions. Oftentimes, these organisms produce a pink pigment. So, not always, but oftentimes when you see a lake and you know it's a salt lake and it has some pink pigment to it, that is because of the organisms that are living in there. Halophiles, for whatever reason, oftentimes produce this pink pigment. And so in very salty lakes, we can oftentimes see this kind of pink coloring because of those organisms. Osmophiles are technically organisms that love osmotic pressure, and most of the time we're talking about sugar content there. There isn't necessarily an exact number that I can give you that defines what is a high sugar content, but think about something like jam or a soda, like a full sugar soda. Those are things that are very, very sugary. And that amount of sugar typically inhibits the growth of anything, including microbes. However, there are some out there, this is especially common in certain types of fungi. Not quite as common with bacteria, but many fungi technically are osmophiles. And they like to grow in these really, really sugary conditions. So if you've ever had like a jam jar that you've left in the back of the refrigerator for a long, long time, it does usually take a very long time. We're not talking about weeks, we're talking about months or potentially years of just sitting there in the refrigerator. And you've seen it come out looking like this, or at least somewhat like that. Maybe the whole thing wasn't covered, but there was a little bit of growth on it. That is because the organism growing there is an osmophile and is capable of growing in very, very sugary conditions where there's not a lot of water present. Once an organism has everything it needs, like the sponge elements, its micronutrients, the temperature is correct, it has enough electron donors, the right carbon source, osmolarity is correct. Basically, once everything is right in its environment, it can start to grow. And if you'll recall from an earlier lecture, the process that prokaryotes use to grow is called binary fission. We talked about this process a little bit in our previous lecture on DNA replication, and essentially the process of binary fission creates identical copies of cells those cells are typically called clones. They can also be called daughter cells, but most of the time in the world of microbiology, they are called clones. So the way this works is that first the DNA is replicated, so the chromosome is completely copied so that each cell that is created in this process can have a full copy of that. The chromosomes are split apart from each other, and once that occurs, the cell can create a ring of Fit-Z proteins, those cytoskeletal proteins, that are used to kind of pinch inward. So it's kind of like you have a big oblong oval shaped structure that has something that's squeezing on the center of it. And that Fit-Z ring will eventually contract to the point that the two cells can split apart, and these identical copies of the original cell have been created. When this process happens frequently enough in a controlled environment, such as a laboratory setting on something like a Petri plate or in a broth culture, something like that, then we can actually start to see the results of binary fission when we start to see colonies of cells form on or in our media. There is a predictable pattern to microbial growth, and we can look at that pattern in a couple of different ways. As cells are going through binary fission, one cell becomes two cells become four become eight, etc. So basically, the population is doubling with every binary fission event. And cultures that are growing go through four distinct stages. The stages are called the lag phase, the log phase, the stationary phase, and the death phase. And each of these stages have what is essentially a unique characteristic or a unique set of events in terms of what's going on with the population number. During the lag phase, the number of individuals is fairly constant. And the reason for that is during this phase, there are relatively few cells, and these cells are not yet going through binary fission. What they are doing in the lag phase is they are taking in nutrients, they are replicating their DNA, and they're getting ready to go through a rapid period of binary fission, but they're not quite there yet. Once they have taken in all the nutrients they need, they've replicated their DNA though, they are ready to go into the next stage, which is called the log stage, or sometimes called the exponential stage. And the reason it's called the exponential stage or the log phase is because from a mathematical perspective, these are growing at an exponential rate. During this stage, the number of cells rises rapidly because binary fission is occurring very, very quickly, and the population is doubling every 10 minutes, 20 minutes, 30 minutes, etc. We'll talk a little bit more about what's called the doubling time of the population on the next couple of slides. But basically, after a given period of time, whether that period of time is 10 minutes, 20 minutes, 30 minutes, 40, whatever it is, the population doubles. Now, populations cannot double forever. Theoretically, that is possible, but in reality, organisms start to use up the nutrients that are available, they start to create waste products, and they start to run out of space. So there is a limit on the number of times a population can double in an enclosed environment. After this very rapid period of growth called the log phase, the next stage that occurs is called the stationary phase. And during this stage, growth slows because the number of individuals that are being produced equals the number of individuals that are dying. So binary fission is still occurring. It may not be occurring quite as rapidly as it was during the log phase, but it is still occurring. So the population is still having new individuals being born or being made. However, at the same time, individuals are dying as well. And in the stationary phase, the number of individuals that are created equals the number of individuals that are dying. So the number of total individuals stays the same during this phase. Finally, enough of the nutrients have been used up, maybe too many waste products have been created, and all of the space has been taken up, and the culture enters what's called the death phase. During the death phase, the number of viable cells, which means the number of cells that are alive and able to reproduce, starts to decline rapidly. While there may still be some binary fission going on and some new cells being created, that cannot make up for the fact that more individuals are dying during this phase. And as a result, the total number of living cells in the population drops dramatically. Now I have a graphic representation of this on the next slide, but I've also created this diagram for you to help to kind of show you what the broth culture would look like if we actually could watch this happening in real time. During the lag phase, we would see a few viable cells here getting ready to go through binary fission, but we wouldn't see a lot of new cells being created. During the log phase, we would start to see a lot of new cells appear in this broth culture. During the stationary phase, we would see some of these new cells growing and reproducing, going through this binary fission process, but others we would see dying, and usually they start to kind of accumulate on the bottom. And then during the death phase, we would see a few viable cells left, but a lot more dead cells hanging out at the bottom there, because basically their life cycle has been completed. So if we look at what is happening during the microbial growth curve from a graphical perspective, this is what we see. We see 4 distinct stages of growth. The first is the lag phase, where the number of cells is just constant, because again, that is the phase where we have some viable cells, but they're not quite ready to divide yet. They're taking in nutrients, they're replicating their DNA, but they haven't quite reached that period of binary fission. So the number of cells is not increasing. The culture is not growing. But eventually they get to that point where they can go ahead and go through binary fission, and the number of viable cells or living cells increases very rapidly. This is the log phase. But eventually, the things like waste products start to build up, the space becomes limited, the nutrients get depleted, and as a result, although there are some organisms that are still going through binary fission and producing new cells, some of them are dying. And the rate at which new cells are being produced equals the rate at which older cells are dying. So the population number flattens out again, because this is another phase where, although we do have some binary fission going on, the death kind of negates that, so the population number stays stable. After the stationary phase, this is a period where there's a rapid decline in the number of viable cells. This is called the death phase. And the reason for this phase is that basically the environment that the organisms are in no longer meets their requirements for growth. It is potentially out of nutrients, maybe the temperature is wrong now, maybe the pH is wrong now, the osmolarity is wrong. Basically, the condition is not conducive to binary fission anymore. So more organisms start to die than are being created. And as a result, we see this rapid decline in population numbers. One final way to look at microbial growth is from a mathematical perspective. We are actually able to use a formula that will tell us how many cells we can expect at the end of a given period of time if we knew how many cells we started with. So this formula might look a little bit complicated, but I'm going to walk you through it and you will be able to do this just fine after we go through the terms and after we go through the example that I give you. What this formula is saying is NF, which equals the final number of cells, equals the initial number of cells, or Ni, times 2 raised to the nth. And the nth equals the generation number. Basically, N here is telling us how many times binary fission has occurred, at least how many generations have occurred, how many sets of parents have been made over time, roughly. So NF, like I said, equals the final number of cells. This is the number of cells that we will end with. And that equals Ni, the initial number of cells, the number of cells we start with. And N is the generation number, whether we're starting on generation 1, generation 2, generation 3, generation 4, etc. That's really basically telling us how many times the culture has reproduced itself. NF is what we are solving for. You will always be given Ni, and you will be shown how to calculate N from the information that is given to you. So NF, that's what you're solving for. Ni will be a given. And then little n, the generation number, equals the time of growth in minutes divided by the generation time. These will also always be given to you. So your job will just be to take the number of initial cells and multiply that by 2 raised to the n. So you will need to figure out little n, and then take 2, raise it to the little n, and then multiply it by Ni. And that's all you have to do. So I'm just going to write out a few helpful terms here on this slide, and then we're going to walk through an example on the next one. So NF is the final number of cells. That's what you're going to be solving for. And that just represents the number of cells there are in a colony after a given time has passed, essentially, whether that time is an hour or 2 hours or 4 hours. It basically is just telling us how many cells there will be in this colony as a result of microbial growth after a certain period of time. So Ni is the initial number of cells. That's the number of cells that we start with in a colony or a culture, and I will give you that. N, little n, equals the generation number, and we find that value by taking the time of growth in minutes, whether that is 30 minutes, 60 minutes, 90 minutes, 120 minutes, and divide it by what's called the generation time. The generation time is the amount of time it takes for binary fission to happen. That's an N, sorry, not an R. So time of growth in minutes is just the amount of time a colony or culture has been growing. And the generation time is the amount of time it requires to actually be able to go through a binary fission event. And what's really interesting about that is that actually that varies quite significantly between different species and also between the same species depending on what type of environment they're in. So just to give you some extreme examples of this, if E. coli is given the absolute perfect environment, then it can go through binary fission in about 5, sometimes 10 minutes. On the other hand, there is an organism called Mycobacterium tuberculosis. You don't need to know that for right now. We'll actually cover that in the disease unit. But it is an incredibly slow grower. It causes the disease that we call tuberculosis, and it usually takes about 30 days for that organism, even under ideal conditions, to go through binary fission. So in a real life setting, the time it takes for a generation to be created or the time it takes for binary fission to happen can vary pretty significantly, but you will be given that value when you're working out an equation. So it's nothing that you'll have to guess on. I will give you that value. And then this last bullet point I have here, 2n equals the number of cells in that generation. Basically what that's saying is just however many cells there were in one generation, this is how we're calculating that value. So you don't have to worry too much about that one, but for some people who are very mathematically oriented, I have found that being able to tell them that or kind of explain it to them in that way, it really helps. So now that you know what the equation is and what each of the terms basically is, let's go ahead and work through an example. So here's our scenario. You went to a picnic and left the potato salad out for 2 hours on a warm day. There were 20 Staphylococcus aureus cells. They have a generation time of 40 minutes in the potato salad to begin with. How many Staphylococcus aureus cells will there be at the end of 2 hours? So we're going to start this out, and I recommend that you guys write it out like this when you do your work too. Start out with Ni. So in this case, that would be 20. And next, we'll figure out little n. Little n equals the time of growth in minutes. So first we want to figure out the time of growth in minutes. So that would be 2 hours times 60 minutes per hour. That lets us cancel out hours and gives us 120 minutes. So we have 120 minutes divided by the generation time. The generation time is 40 minutes. So our little n equals 3. That's how you figure out little n. The next thing we're going to do is start putting these values into the equation. So NF, I've already got that written for you. That equals 20, which is the number of cells initially, times 2 raised to the third. The next step you want to do is do 20 and then figure out what 2 raised to the third is. Remember, when we're doing exponents, we're not doing 2 times 3, we're doing 2 times 2 times 2 in this case, or if you had a generation time of 4 up there, it would, or generation number of 4 up there, you would have 2 times 2 times 2 times 2. So just if you need to review exponents, definitely do that. And if you need help with them, let me know. But basically we have 20 times 8, because 2 raised to the third is 8. And so then when we look at our equation, we just take 20 times 8, and that gives us an NF of 160 cells. So what this is telling us in words is basically that there were 20 cells to start with, and those cells were able to go through binary fission every 40 minutes. And after 2 hours, 3 generations of cells were able to be created. As a result of that, our 20 initial cells end up turning into 160 cells total. So I know this equation can be a little bit intimidating at first for some people, especially if they were not expecting to have to do a lot of math in the class. However, I promise you, you will be able to do this. I can create some additional practice problems if you would like me to, or we can just work on those through email and I can assist you with them. But in the end, I have found that anyone who has at least a little bit of time to work on this and is interested in figuring this out is able to do so. So far, we've gone over a lot of different concepts in this lecture, a lot of pieces of information, and to give you a big picture perspective about why we care about this, we're going to talk about 2 concepts related to microbial growth that will hopefully help you see why this matters so much, especially to pre-healthcare providers. Really, it kind of matters to everyone in the world, but especially to people going into the healthcare professions. This may give you a different perspective on why understanding microbial growth is so important. In certain cases, when we are dealing with a patient or a person who has an infection by a bacterium, it can be very helpful to understand what phase of growth those microbes might be in, because oftentimes antibiotics, or the medications we use to treat bacterial infections, work better when the organisms are in the log phase. So it might seem a little counterintuitive, but during the log phase, bacteria are very, very metabolically active, which means they're taking in a lot of substances from their environment. So if we want to make sure that a certain type of antibiotic is going to be effective to treat an infection, it is very helpful to know what stage of growth these organisms might be in, because that can determine how quickly the organisms will take up the antibiotic from their environment and how quickly the drug will work to kill them or at least stop their growth. Another concept that is related to microbial growth that is significant is this concept of infectious dose. So infectious dose is the number of individual cells or particles of a microbe that it takes to make a person sick. We don't always know the exact infectious dose, but when we study a microbe, we can determine what's called the infectious dose 50, or the ID 50. And basically the ID 50 is, like I said, the number of cells that it takes to cause an infection or to make people sick in about half of the population. So just giving an example here, let's say the ID 50 of a certain microbe is 100 cells. That means if you line up 100 people, and you give each of them 100 cells of this microbe, 50 of them will get sick from it, because that will be enough microbes to cause disease in 50% of the population. The ID50 is important and significant in a lot of scenarios. You may have even heard about ID50 related to COVID in terms of how many particles of the virus does it take to actually make someone sick. But one especially significant example of ID50 is when we're looking at foodborne pathogens. If we are trying to evaluate whether or not a food will be safe for consumption, we have to be able to determine how many microbes are in a colony that's in food at any given point in time. So the reality is most of the food we eat is not free of microbes. Most of the food we consume has a certain number of microbes in it. But most of the time, there are not enough microbes in that to make us sick. But if we're talking about food safety overall in something like a kitchen or an industrial setting or food production, we need to be able to calculate the number of microbes that were in a certain type of food to begin with, how much time has passed, how long the generation time is, and how many microbes might be in a certain food at a certain point in time to determine whether or not that food might be safe for people. So being able to understand microbial growth is significant in a lot of settings, but two of the particularly significant settings are understanding how and when certain medications might be most effective when they're given to a patient, and also, especially in food microbiology settings, whether or not a particular food or particular organism, whatever we're looking at, is going to be safe for consumption depending on how much time has passed after that food was manufactured or created or cooked.